Good Monday morning, everyone. Welcome to week number 5 in Biology 120. It is also Thanksgiving week, so I know it's going to be a busy one for you. I know that I'm cooking for about 20 people, so yeah, it's going to be a very busy week for me too. I hope that you have a chance to spend some time with family and friends and celebrate this season. But you do have some work to do, so let's get into that. All right, so last week you learned about metabolism, which is all the chemical reactions that happen in an organism. You learned about catabolism, which is the breakdown of food to produce energy and the small units of molecules that you need in order to build your own, and anabolic processes, which are the building up of molecules that you need. And we learned about different types of catabolic reactions: aerobic respiration, anaerobic respiration, and fermentation, and you did some discussion of that. We also touched on photosynthesis, which is the process where carbon dioxide, with the use of sunlight, is converted into glucose molecules by many organisms on the planet, and those are called autotrophs, and autotroph means self-feeder. So all those photosynthetic organisms feed themselves because they make their own food using sunlight. Okay, this week is all about DNA, which is, of course, one of my favorite subjects. Where's my DNA molecule? DNA, I love it, love it. All my research was on DNA, and I'll be talking about that in a second. Parts of it that I'm very, very interested in. So this week, at least skim through chapters 10, 14, and 15 in the textbook, your OpenStax textbook. Chapter 10 is all about the cell cycle, which I'm going to mention right there in my picture. Chapter 14 is about the structure of DNA, and 15 is what the purpose of DNA is. So let's touch on those for a second. The cell cycle describes the life cycle of a cell, and this is a representation of it right here to the right. And you see all these G's and letters and slashes. Okay, so a cell cycle is just that. It's a circular life cycle, like so many are. And even if you're talking about other types of cycles in nature, like the carbon cycle, what happens to carbon dioxide is it moves through the environment and then back up into the atmosphere. So we have a lot of cycles in biology. One of those is called the cell cycle. So you start at the 12 o'clock position, right? That's when a cell is newly made. And then you have some G's and S's as you go around. So what does G stand for? G right now stands for growth. It's a part of the cell cycle where there's growth. That's not really what it stood for when we started describing the cell cycle. G stood for gap because we didn't know what was going on. But as we've studied cells, now we understand this. So we call it growth. S stands for synthesis. That means you're making something, okay? So let's start up at the 12 o'clock. Then we have G1, and part of G1 is G0. So G1 is general growth of the cell. So it's the increase in size, it's an increase in the number of mitochondria. Of course, we're talking about eukaryotic cells here. There's an increase in lots of proteins that are needed for the cell to get bigger in order to divide. Just general growth. All right, now I say that G0 is a part of G1, and that is because a cell's job is not to divide. A cell has a particular job in life to do, whether it's a single-celled eukaryotic organism or a heart muscle cell of yours or a liver cell or a cell in the small intestine. They each have jobs to do. And that's what G0 is. There's a stopping point in G1 where the cell has grown enough where it can actually stop and do its work. That is G0. Now, the importance of this is that cells stop in G0, except cancer cells. Cancer cells never stop in G0. They just keep going round and round and round and round the cell cycle, dividing and dividing and dividing, and they never stop. So that's an important point about cancer cells. Okay, so we've got G1, the cell's grown enough, then it shifts into the S phase of the cell cycle. S, remember I said, was synthesis. This is where new DNA is produced. So DNA replication, and that is part of chapter 14, I think. Anyway, synthesis, the production of new DNA, and DNA has to be copied in order to have good copies of DNA moving into the two new daughter cells when this cell divides. Then you've got G2. So what happens in this second growth phase? This is where you make all of the cellular machinery that's needed for cell division. The spindle fibers, the centrioles reproduce, etc. So all the cell machinery needed for cell division is produced in G2. And then you have M and C. M is either mitosis or meiosis. That's a type of nuclear cell division where you separate chromosomes from each other. And C is cytokinesis. All the rest of the cell divides. So even the cytoplasm, the liquid portion of the cell, divides in cytokinesis. And then you have that 12 o'clock noon position again, because that's the beginning of this brand new cell. So that's the cell cycle. There are a couple of other important points here. There are checkpoints all along the way where the cell will stop if certain things haven't happened. So in G1, if the cell has not grown large enough to divide, if it doesn't have enough mitochondria to support two daughter cells, it will stop in G1 at this checkpoint. There are checkpoints all along the cell cycle where the cell stops and evaluates to see if it's ready to move on to the next phase of the growth cycle. And normal cells do this. They stop when there's a problem. Once again, with cancer cells, they don't adhere to those checkpoints. They just keep on dividing and dividing and dividing and dividing. And so you get all kinds of mutated cells when you're talking about cancer being produced in someone's body. And one other problem is they don't ever stop dividing. If we could figure out how to regulate their cell cycles, we could stop cancer. And this has been studied since the 1950s, and we still don't have the answer to this, but we do have many new good therapeutics that treat cancer cells now and cancer in the body. Okay, also read the chapter 5 overview and watch the videos. Those videos will help you understand DNA replication, the cell cycle, the central dogma of molecular biology. That I wanted to also mention. So the job of DNA is not to reproduce. The job of DNA is to make all the proteins, and remember, most of those are enzymes that a cell needs. And that's the central dogma of molecular biology. DNA is used to make a small piece of messenger RNA, which is then used to make a protein. So the job of DNA is to make all the enzymes that run all the chemistry in cells. Okay, the discussion board postings this week are about cancer and carcinogens. And this all has to do with not only DNA and DNA replication, but also the cell cycle. Okay, so let's get into it. You're talking to a friend and you're having an argument over whether cancer is a problem with mitosis, which is the nuclear division of the cell reproduction cycle, or is it a genetic disease, meaning is it found in the genes? Or, oh, here we go again, both. What did I tell you last week? If you ever see both, you know that the answer is both. So is it a problem with cell division? I just explained to you over there with the cell cycle that cancer cells never stop in G0 and they do not adhere to the checkpoints. They just ignore them and just keep on dividing. Is it a genetic disease? Well, I bet you've heard of genes that are involved in cancer production. Just two examples would be the BRCA genes, the breast cancer genes 1 and 2. that put you at much higher risk for developing breast cancer and or ovarian cancer if these genes are present. So I know that you've heard of these before. So you're going to look up some information about these, and you're going to find out what proto-oncogenes are and tumor suppressor genes. These all have to do with cancer production in your bodies. Okay, and then you're also going to mention how lifestyle changes can lower your risk for developing cancer. Nothing will make your cancer risk go down to zero, because just living means that you're going to have mutations that build up in your cells that could lead to cancer. That's just a part of living. But what lifestyle changes can you do to lower your risk? Okay, and then in your response post, you're going to talk about environmental carcinogens. A carcinogen is any type of mutagen, causes mutations, that produces cancer. So there are so many, so many. I'll give you one example. On our gas stations here for a long time, there used to be a little sticker on them that said, "Don't breathe in gasoline fumes because they contain benzene." And benzene is a known carcinogen. Those stickers are gone now. Not sure why. Don't guess the gas companies wanted you to read that. But anyway, benzene is an environmental carcinogen which you should not be breathing in. How do you remove these from the environment? There are lots of different ways. One is to not breathe in the fumes, which is why that sticker was there. But anyway, the stickers are gone now. So how can we remove them from the environment? And there are so many of these. You will find plenty when you just do a Google search of environmental carcinogen. I promise you. All right. And so you've got the discussion board postings. Remember, the initial post is due by Thursday night. The 2 response posts are due by Sunday night. Then you have milestone 3 of the final project. The last part of your final project before you put them all together, all 3 of the milestones, in week number 7. Next week you're going to start on this though, right? Okay, deal. So the final part of your final project, this milestone 3, is how to fix this problem or issue that you've been discussing. So here are the sections. Methods of conservation. This, if you will think about this as what are some ways that we could prevent this thing from happening? How can we prevent water pollution from occurring? How can we prevent eutrophication? How can we prevent global warming, climate change? Okay, that's really methods of conservation. Education. Why is education about this topic important? That's always going to be important. I don't care which topic you're talking about. Solutions. How do we fix this problem? So methods of conservation is how to prevent. Solutions are how to fix the problem. How can we fix the fact that we have dumped so much carbon dioxide into the atmosphere from the burning of fossil fuels? Good question there. Plant some more trees. Hello. Anyway, there are other solutions. Ethical considerations. And in this section, don't forget about laws, regulations, guidelines that are already in place that help this particular issue that you're discussing, or laws, guidelines, or regulations that could help this particular situation. Then you're going to discuss positive and negative aspects of the issue. Usually there is always negative aspects, because that's why this is a topic or a problem or an issue. But are there any positives? You really need to think through that. So with global warming, for example, you know, there are lots of companies that are benefiting from the sale of fossil fuels, right? So there's a whole huge industry behind this, and they don't want to stop doing what they're doing. All right, and then personal impact. What can you yourself do about this particular situation? How can you change your behavior to help this particular topic be solved. Include citations, in-text citations, give people credit when you get information from somewhere, and a whole reference section. In milestone 3, you have to have at least 3 references. So remember that. So you're going to have quite a few when you get to your final project. This time, the format is PowerPoint slides. You don't have to add speaker's notes, and I'm going to talk about that in week 6. You don't have to have a speaker's notes this week, but you will for the final project. And label all of your sections. I cannot emphasize this enough. Methods of conservation should be at the top of the slide so that I know that this is what you're talking about. The problem is when you just write this essay style and you just write on and on and on and on and on, I don't know that you know that you're answering these questions about your topic. So please include the section labels, the headings, and I have them listed right there in 5A, okay? All right, this last part is just interesting to me, so if you want to keep watching, that is awesome. If you don't, that is perfectly fine, because this doesn't have anything to do with your assignments. But it is one of my passions. DNA fingerprinting, also called DNA typing, DNA profiling, there's lots of names of this. So this is the use of DNA in solving crimes. I have been interested in this since the 1980s when I first heard about it. I did DNA fingerprinting for part of my dissertation. I used it afterward to help an oncologist that we worked with. I've taught it to students throughout the years, and I'm still interested in it now, and things are changing now, which is very, very exciting. So how did this come about? In 1985, a professor of genetics called Alec Jeffreys in England was studying DNA. And he was studying DNA in a very closed society. So the members of this society were very related to each other, if we can say it that way. And he noticed that if he chopped the DNA up in a certain way and looked for variable regions, so places where DNA is different in individuals, that he could actually tell the difference between the DNA of one person and the DNA of another person. He also could tell who was related to who. So he could look at a mother's profile and a father's profile and figure out if this child belonged to the two of them. Okay, so he developed this DNA technique, which is called RFLP, and we won't get into that. But he developed this technique, and he went home and he told his wife, and she said, you know what, that sounds sort of like fingerprinting. So that's where the terminology came. DNA fingerprinting doesn't have anything to do with fingerprints. It just means it's a way to profile you and to identify you. So he published this work, and the police in England read about it. They said, ooh, this could come in handy for solving crimes. And they had a set of 3 murders of some teenage girls, rapes and murders of 3 teenage girls in 3 villages in England. And they had no suspects, none whatsoever. Nobody had seen anything. So they had no idea how to solve this. So they went to Alec Jeffreys and they said, listen, can you use that DNA fingerprinting technique and analyze the seminal fluid that we've collected and help us figure out who murdered these 3 girls? And he said, I think so. Let's try. So they did. And he analyzed the 3 rape specimens. And sure enough, they were all from the same person. So the police in England said, okay, we're going to collect blood from all the men in the 3 surrounding villages, and we will bring those samples to you, and you check them and see who is our suspect here. Alec Jeffrey said, well, that's a pretty big project, but okay, let's go ahead. Civil liberties aren't the same in England as they are here, and so the police just required men to come in and give blood samples. And they did. And they collected over 5,000 blood samples from the men in these 3 villages. Now let's think about this for a second. If you are the perpetrator and you hear about this new technique, are you going to willingly go in and give your blood? Nope. And that is what happened. A man named Colin Pitchfork, I'm not kidding, that's his real name, went to his friend Ian Kelly and he said, hey, listen, I've been sick and I really can't give blood. So how about you go in and give blood a second time for me? So Ian Kelly was convinced that this would be a good idea, and he did. Meanwhile, later on in the day, Ian Kelly got drunk at a pub and told everybody what he had done. And one of the women there said, this just doesn't sound right. So she told the police. And they went and they grabbed Colin Pitchfork and got blood from him. In the meantime, Sir Alec Jeffries, he wasn't Sir, he hadn't been knighted then, but Dr. Alec Jeffries was testing some blood from someone who had confessed to the crime. And he went to the police and said, nope, this is not the person. So the first time DNA fingerprinting was ever used in a criminal case exonerated someone who was innocent. In the meantime, back at the station, they brought Colin Pitchfork in and they have collected his blood. Alec Jeffries runs Colin Pitchfork's blood, and sure enough, he matches the evidence that they have from this case. And he was convicted based on this, and he is spending life in prison for it because he did do it. He was absolutely not a suspect. He was a married man with a job, owned a house, had some children. There was no idea in this world that he would have perpetrated these 3 rapes and murders. So, of course, this made big news. And I mean big news all around the world. This was 1985 over into 1986. By 1988, the FBI started using this technique in the United States. So it started with the FBI, not with the state agencies, but then when the state agencies heard about this, state police agencies, they started opening DNA labs. And besides that, we have even local DNA labs in different counties. I know I have one in my county. So they are everywhere now. They do not use the same technique as Alec Jeffries did because that was the RFLP technique. And it needed undegraded DNA. It needed all of the DNA from cells. And this is usually hard to come by when you're talking about evidence in criminal cases. So another technique was developed using the same idea, though, looking for those variable regions in the DNA. And now we use something called PCR. You've heard about this because PCR is one of the types of tests run for COVID. PCR is the polymerase chain reaction, and what you're doing is just making copies of DNA in certain little segments of the DNA, and those would be in those variable regions. So this type of technique can use very degraded DNA, and a lot of evidence in criminal cases is very degraded. It's been decomposing for a while. And you don't have to have all of the DNA. You just have to have these little tiny sections where they're variable regions. We're having new technologies being developed now. There will be handheld DNA testers that will be on the market soon. So you'll be able to do this out in the field, which is very interesting. Also, something else has happened. We had a lot of websites being set up to trace your ancestry. These genealogy websites have been used by police to catch criminals because they will submit the PCR results from some evidence, and they're looking then to see if there are any matches out there for this DNA. Now, usually there's not a one-on-one match to one particular person, but they can trace this DNA into certain families, and then by elimination, they can figure out who might possibly have contributed this DNA, and then go out and collect the DNA from that person, test it, and see if that person matches the DNA. This particular technique was used to catch the Golden State Killer, who had been on the loose for over 40 years. His name is Joseph D'Angelo. He has not been convicted yet, but it's a given. His DNA matches these horrific crimes in California. For a long time, he first broke into houses. He raped women. Then he started murdering the people in the houses, and he was just a horrific criminal. He would break in on purpose to a house that had a man and a woman inside. He would tie up the man and put cups and saucers on the man's back so that he could hear if the man got up, and then he would go in the next room and rape his wife. So it was horrific. It was just psychological torture that he was doing. And he was caught. He was identified through genealogy websites and turning in DNA from the evidence. This is going to be more prevalent. You hear about it really on a monthly basis now. They're catching more and more people, or identifying who did a crime even back decades ago. Even if the person is dead, even if the perpetrator is dead, they're being able to figure this out right now. So this is a cool area of research. It's an active area of use these days in using DNA to catch criminals. If you have any questions about this, or if you're interested in it, I would love to talk to you about it, because of course it's one of my passions. So anyway, I hope you have a wonderful Thanksgiving week this week. I hope that you get a chance to unwind a little bit and celebrate with family and friends. And as always, if you need anything, there's my name down there, Dr. Janie Sigmon, m.sigmon@snhu.edu. Just shoot me an email and I will answer it. Have a wonderful week.